It's a pleasure to be with you virtually. The title of my talk is How Can We Help the Damaged Kidney to Regenerate? These are my disclosures. They are not relevant to what I'll be presenting today. I'll be touching on three different topics relevant to AKI. The first is how the use of kidney organoids is informing our approach to modeling AKI in the lab and potentially treating it clinically. The second is targeting a metabolic switch using niacinamide in a strategy that is much closer to clinical application. And finally, I'll provide a broad overview of the Kidney Precision Medicine Project, which utilizes recent technological advances to understand AKI at a much finer resolution than what's been possible before. When a kidney undergoes an acute injury from any cause, there is tubular damage and tubular obstruction, leukocyte recruitment with inflammation, endothelial damage, and activation. In many cases, the repair process, which is shown on the far upper right, proceeds normally with resolution of inflammation, epithelial proliferation, and repair and regeneration of the vasculature, leading to reconstitution of kidney function. In other cases, however, repair does not proceed normally, and inflammation persists, leading to the accumulation of scar tissue in a process known as maladaptive repair. This can lead clinically to chronic kidney disease and in some cases end-stage renal disease. The therapeutic approach then for AKI is to promote or accelerate productive repair on the upper right and to inhibit maladaptive repair on the bottom right. Anytime one thinks about therapeutics for human AKI, though, it's important to bring a healthy dose of humility given the many failures clinically over the last several decades. Here are two more recent examples. On the left, the START-AKI trial, which examined early initiation of renal replacement therapy in ICU patients suffering from AKI compared to normal. The difference was about 12 hours, but as you can see, there was no difference at 90 days in survival between these strategies. A second recent negative trial is shown on the right, the PRESERVE trial, which compared the use of sodium bicarbonate with intravenous sodium chloride and N-acetylcysteine versus placebo in preventing radiocontrast-induced acute kidney injury. This was a well-powered, well-designed trial that, again, as you can see on the far right, delivered completely negative results. Despite this, we're all familiar with the very encouraging results from clinical studies examining the use of SGLT2 inhibitors to slow progression of diabetic nephropathy. Here shown the CREDENCE trial with extraordinarily positive results with the use of canagliflozin regarding kidney endpoints. This kind of result, which has been replicated in other trials, gives us hope that we might be at the dawn of a new era with new therapeutics on the horizon for treatment of kidney diseases in our patients. I'll now focus on kidney organoids. This is a relatively new field. As you can see on the left, the very first papers describing it were published just about 7 years ago. The concept here, though, is relatively straightforward and is shown on the cartoon on the right. We now have the capability to take somatic cells from an adult patient and to reprogram them into pluripotent stem cells in the lab. We can then, in a stepwise differentiation protocol, differentiate them into little mini kidney organoids in a dish. These can then be used to model kidneys on a chip for transplantation into mice. And ultimately, the goal is to use them as replacement tissue for patients that have lost kidney failure. The lay press became quite excited when these results first were published with headlines such as "Lab Kidneys a Step Closer," "Transplant Breakthrough, Kidneys Grown from Stem Cells." But then they became a little bit more unrealistic. "Growing kidneys in the lab may end the transplant wait list, and kidney breakthrough could end the need for donors." I assure you, this is not anywhere near where the field stands. But let me show you some of the things that we can do with organoids. On the left, I'm comparing a healthy adult real human kidney section to sections of a kidney organoid grown in the lab. If you focus your attention to the bottom right, you can see that the lab-grown organoid actually has clear glomerular-like structures surrounded by tubules, and the overall architecture is very similar to what you see in the real thing on the left. The primary difference being the enhanced nuclear to cytoplasmic ratio in the organoid, reflecting the immaturity of those cells. By scanning electron microscopy, we can see here a proximal tubule, and if you focus on the bottom right, it's easy to see the microvilli that are a characteristic of proximal tubule cells that are helped to be responsible for the transport properties of this particular cell type. And it looks very much like what we would see in a real human kidney. Moreover, these kidney organoids can respond to injurious stimuli in ways that are similar to the ways that human kidneys do. Here, the tubular toxicant cisplatin has been given to a dish containing kidney organoids, and on the left you can see there is a brisk upregulation of tubule injury markers, NGAL and HAVCRA1, which is also known as KIM1 or kidney injury molecule 1. On the right, you can see that these protein markers are upregulated by immunofluorescence in red in the appropriate tubular segment, proximal tubule for KIM1 and distal tubule for NGAL. Thus, kidney organoids make an outstanding model for human AKI, getting us away from rodent models, which we have been dependent on for so long. More and more, we are able to study kidney functional responses in organoids as well, and that is what is shown in this picture here. On the left, a cross-section of a collecting duct with aquaporin 2 in red. Aquaporin 2 is the water channel that responds to vasopressin, and when serum osmolarity rises, AVP is secreted by the hypothalamus, travels to the kidney, and in the collecting duct causes aquaporin 2 to be inserted into the luminal membrane. And that's what you're actually seeing here. On the top is a collecting duct without arginine vasopressin, and on the bottom we've subjected it to 3 hours of DDAVP. And I hope you can see that red stain has now translocated to the middle of that tubule, indicating that this signaling loop is active within our kidney organoid. Despite these impressive advances over a relatively short time period, kidney organoids are not near ready for clinical translation for several very important reasons. Number one, they contain cells that are not normally found in the kidney. For example, on the bottom left, you can see in green and yellow, there are neurons that are hiding out in the interstitium of kidney organoids. Other cell types can be found, for example, muscle. These cells make up substantial proportions of the total cell number in organoids, up to 20 or even 30%. There's also a lack of vasculature. In green, you can see endothelial cells, but they are not linked together and they do not form a lumen, nor are they perfused with blood. Kidney cells in general, as I alluded earlier, are also quite immature. They resemble the second trimester gestation, not an adult level of maturity. And finally, there's a plumbing problem with kidney organoids in that there is no collecting system. So even if we were able to develop a vasculature and have blood flow through it and generate a filtrate in the glomeruli, it has nowhere to go. The picture on the right is a cross-section of the collecting duct showing, again, principal cells in green and intercalated cells in red. But the point here is that this ends in a blind pouch. It does not hook up with other collecting ducts. It does not empty into a ureter, which connects to a bladder, so that if we transplanted these organoids into a suitable mouse and developed a filtrate, it would result in a fluid-filled cyst and would not empty. For these reasons, then, organoids have great promise in the preclinical realm, but they are not ready for clinical translation. By contrast, targeting the metabolic switch that occurs in acute kidney injury and proximal tubule is a very promising therapeutic strategy that is already in clinical trials. And that's what I will describe in these next few slides. I want to provide a brief refresher of energy metabolism in proximal tubule. The proximal tubule can create energy in the form of ATP by one of two methods. On the left, by glycolysis, in which NAD serves as a universal electron acceptor and creates two ATP molecules for every molecule of pyruvate. This is a process that does not require oxygen. On the right, in fatty acid oxidation in the Krebs cycle, 36 ATP are created for every molecule of pyruvate. This is a much more efficient process, and indeed it is the process that the proximal tubule relies on predominantly. Both fatty acid oxidation and fermentation and glycolysis require NAD. Now, in acute kidney injury, it has become clear through a variety of studies over the last several years that damage to mitochondria lead to a metabolic switch from fatty acid oxidation to glycolysis, which does not require oxygen, but it does require NAD. Now, in work that has largely been completed by Samir Parikh in Boston, he has uncovered an important deficiency in the biosynthesis of NAD, which is rate limiting, in AKI as well as this metabolic switch. Now, here are the pathways for synthesis of NAD. On the left, the de novo pathway, in which tryptophan is metabolized to a precursor, quinolinate. Quinolinate can then be generated through the action of an enzyme, QPRT, into NAD, and this is the predominant pathway by which NAD is generated in the proximal tubule. There is a salvage pathway, which is shown on the right, in which NAM, or nicotinamide, can be converted to NAD, but the proximal tubule does not rely on this pathway. Now, it turns out in AKI, quinolinate builds up, and this is what's shown in red on the upper right graph. That suggests a deficiency in QPRT levels, and indeed a deficiency in NAD levels, which is shown on the bottom right in AKI. That hypothesis has been shown to be true. Indeed, in this study on the left, a QPRT enzyme was knocked out genetically, and AKI was induced in laboratory mice, and you can see in the red bar that the severity of AKI was enhanced with QPRT deficiency, which also led to a reduction in NAD levels. Importantly, this phenotype could be rescued by exogenous administration of NAM, which presumably through the salvage pathway could rescue NAD levels, and that's shown on the right-hand red bar graph. NAM can also be given in a laboratory model of AKI in a therapeutic dosing strategy after the injury, and that's what's shown on the right. In red, when NAM, nicotinamide, was given, you can see that the levels of creatinine rise were significantly blunted compared to placebo. These laboratory studies have been extended to small human pilot studies, and that is what is shown here. On the left, the experimental protocol for 3 different groups of ICU patients who were scheduled to undergo on-pump cardiac coronary artery bypass grafting, a procedure with a high incidence of AKI. There were 3 groups, a placebo, a low-dose nicotinamide group, and a high-dose nicotinamide group. In the upper right, you can see in the light and dark blue that the nicotinamide administration did successfully raise serum NAM levels, and that in patients who received either the low or the high dose of nicotinamide, there was a substantially reduced, in the bottom right, incidence of AKI in red amongst participants, about 15 or 20% in the nicotinamide group compared to almost 50% in the placebo. So this is an encouraging proof of principle. Here is a more recent study related to COVID AKI that just came out actually several months ago. This was a prospective, non-random observational study of COVID AKI, 201 patients from 2 hospitals. Both of these hospitals had identical COVID care algorithms, but one site administered nicotinamide to COVID AKI patients and the other didn't. And you can see that the renal replacement-free survival of all patients on the left, or of just the more severe AKI patients on the right, was substantially improved in the red line, the group that received the nicotinamide. This was associated with a lower risk of renal replacement therapy or death over the 30-day period. Now, again, this is a prospective, small, non-random trial, but these kinds of results, when combined with the biologically plausible mechanism, have led to the development of several larger prospective randomized blinded controlled trials, 3 of which are summarized here. The Nirvana Trial, which is an expansion of the COVID AKI pilot that I just described, the NID augmentation in cardiac surgery associated myocardial injury trial, or NACAM, and the BASIS trial. Stay tuned for results from these studies in the next 3 to 4 years. Finally, I will touch on the application of exciting technological advances to better understand AKI and generate novel therapeutic targets, as exemplified by the Kidney Precision Medicine Project. This is a consortium that is funded by the National Institutes of Health that really has as a goal of bringing new therapies to diseases like AKI and chronic kidney disease, which sorely need them. This is a multi-year collaboration planned to be 15 years. It's currently in year 4 of leading research institutions, as I mentioned, to better understand basic mechanisms. A high-level overview includes a patient engagement strategy and a tissue biospecimen strategy, particularly in AKI, which we don't usually biopsy. These samples are sent to a central biorepository and then distributed to various tissue interrogation centers where very cutting-edge technologies are used to generate very large data sets, which can now be analyzed using computational approaches like artificial intelligence, ultimately generating human kidney AKI atlas. The kinds of technologies that are being brought to bear are summarized in this slide here. They include single-cell RNA sequencing, which I'll show a very brief overview of, proteomic approaches, metabolomic approaches, revolutionary advances in imaging, and even epigenetics. As I mentioned, this consortium is being driven by technological advances, and one of the most impressive ones over the last 5 years has been our ability to measure thousands of genes in one cell across thousands of cells. And this is made possible by microfluidic technology. This movie depicts just that technology, in which that black bead coming from left to right is covered with DNA oligonucleotides. And there's a very small cell that comes in at the bottom right now that gets co-encapsulated with that bead. The cell releases its mRNA, which is bound to the bead, and through next-generation sequencing technologies, we can generate data sets that look like this, which is a single-cell atlas of acute kidney injury. Here, 125,000 cells from kidneys over a full acute kidney injury time course have been analyzed, each cell with 3,000 different genes measured. And the depiction on the left is a visual representation of these cells. The same color represents the same segment, so you can see up at the top proximal tubule. Each dot represents a single cell, by the way. We can see leukocytes, endothelium, podocytes, injured cells, and generate maps of the single cell response of acute kidney injury. And in this way, when combined with some of those other exciting technologies, hopefully generate novel hypotheses and therapeutics for the next generation of targets. In conclusion, then, kidney organoids have substantial long-term promise, but at least for the next several decades, I believe that's limited to the preclinical setting and modeling of AKI, certainly not as a source of replacement tissues. On the other hand, our understanding of the metabolic switch in AKI and how NAD levels can be manipulated through exogenous nicotinamide supplementation is highly encouraging and has a biologically plausible mechanism. Finally, advances in massively high-throughput methods, such as single-cell RNA sequencing that I briefly provided an overview of, combined with enormous advances in computation, offer extraordinary promise for the future, as exemplified by the Kidney Precision Medicine Project. Thank you very much for your attention.